So this video will cover the extra features of this program, and that will be the 3 optional flags that you see here. And once I've explained that, I'll talk about how to use them in the command line interface and the graphical user interface. Don't forget to check the description down below for timestamps and for all of the links that are mentioned in this video. So this is the first optional flag that we're going to talk about. This part will cover what is the VEP file and how do you create it and what does it look like. Alright, so I'm going to run this so you can see the help manual. So at the bottom over here it says optional flags, and one of the optional flags that we're going to first go through is the --vep-file, or you can also use -v. So we're going to talk about what that is and how we can use this. So if we go to the checklist, you can see we have chromosome number and position, ID, reference allele, alternative allele, ancestral allele, CAD/FRED, and the allele frequency. So some of these columns are mandatory VCF columns. So I'm going to go through what that means. So here is an example of a VCF file format, and this is the VCF specification document. And if you scroll down, you can see that these 8 headers are mandatory in a VCF. So you would have the chromosome and position, ID, reference allele, and alternative allele. So all of this 5 information, as we mentioned in the checklist, are all mandatory. The ones that are not mandatory are the ancestral allele, CAD/FRED, and allele frequency. It is possible for you to come across a VCF file that has the ancestral allele, CAD/FRED score, and the allele frequency for each population annotated in the info column. So I'm going to show you what that would look like. Okay, so this would be all of your mandatory columns, including the format column over here, but I'm just going to focus on these columns. So the info column can contain different types of annotations, and this would depend on the person that is making the VCF file. So it is possible that you can find the ancestral allele, the CAD/FRED score, the allele frequency for each population in the info column. But if you can't find them in the info column, then there are ways to determine what the ancestral allele, the CAD/FRED score, and the allele frequencies are for each population. So first we're going to go through what to do if the allele frequencies for each populations are not annotated in the info column. So if we go to the wiki page where it says extracting information that is not annotated in the VCF file, you can see the first thing that we mention is the allele frequency. So if the allele frequency is not in the VCF file, we suggest using the BCFtools fill tags plugin. So I'm just going to open that link so you can see. So this plugin can calculate the allele frequencies for each population, and how you would do that is you would provide a file with the list of samples. So that will be in one column. This would be the individuals that you have in your VCF file, and then in another column, and this is a tab delimited file, and in the next column you would mention what population they belong to. So over here you could have pop1, but let's say sample 3 belongs to pop2. And then what you can do is then use the BCFtools query command to extract all of the information, including the allele frequencies for each population. So this would be the command here. So to recap, if you want to find out the allele frequencies for each population, you can use the fill tags plugin. Next, we're going to cover what to do if the ancestral allele or the CAD/FRED score is not annotated in the info column of the VCF file. So I'm going to go to the wiki page, and we actually cover that in the next part. We recommend using Ensemble VEP, and VEP is short for Variant Effect Predictor, and it is a software that can grab annotations from databases. So to annotate the ancestral allele in the CAD-FRED score, the VEP relies on two plugins, the ancestral allele plugin and the CAD plugin. So we're going to go through that right now. So I'm going to open this link so you can see. So this is the page for the variant effect predictor, and it has a command line interface which can be used to annotate large files. So if we scroll down here, it says existing plugins, and we'll just open that. You can see that the first plugin that shows up is the ancestral allele plugin, and this is the plugin that we spoke about earlier. So let's just click more so you can see. So this VEP plugin retrieves ancestral allele sequences from a FASTA file, and you can download the FASTA files from this link here. So there are several versions out there for each reference genome, and once you find the version that you want, you can then follow the instructions over here, which basically processes the FASTA files into one file that is bgzipped. Next, we're going to talk about the CAD plugin. So this CAD plugin is the one that we referred to here. So this VEP plugin retrieves CAD scores from their data files. So their data files consist of the CAD-FRED scores of all of the alternative alleles in the reference genome. So you can download the data files on their website here. So I'm going to copy that so you can see. Okay, so this is their latest version, and it's version 1.6, and they have other previous versions as well. So for this version, they have two data files. One of them is for HG38, which is this one over here, and the other one is for HG19. And then what you can do is type in a command similar to this in order to annotate the ancestral allele and the CAD-FRED information. So to recap, if you want to annotate the ancestral allele, you would use the ancestral allele plugin. If you want to annotate the CAD-FRED score, then you use the CAD plugin on VEP. And you can actually run both of them in one command so that you can create one output file. So the VEP file flag is used to supplement information like the ancestral allele and the CAD-FRED scores. So if you're using the command line interface, you can specify the VEP file by doing this. Let me just open up the manual. So you can specify it by either using -v or --vep-file and then specify the file. If you're using the graphical user interface, you can specify the VEP file here, where you would click Browse and then select the file. So how would you make the VEP file? So we have an example here of what a VEP command would look like if you were to annotate the ancestral allele and the CAD-FRED information. So here you have the VEP, then you would specify the input file, which is your VCF file, and then we would have the two plugins that we spoke about. So you can rearrange this in any order. The first one would be CAD, and then you would specify the data files. So data files contain the CAD-FRED scores for all of the alternative alleles. And then the next plugin would be your ancestral allele plugin. And over here would be the processed bgzipped file. Next, we want to output this as a tab delimited file. So to do this, we would use this flag here called the tab flag, and then this flag here, which is the fields. So if we open the VEP manual, you can see that this flag here would write the output as a tab delimited format. And over here, where it says fields, you can specify the fields that can be generated in the output file. Okay, so going back to the wiki page. So over here, we have the fields location. So location is the chromosome and the position. Then we have the uploaded variation. So this serves the same purpose as the VCF's ID column, where you would have the identifier. So this field is optional. Either you include the ID from the VCF file or this uploaded variation. Next, you have the ancestral allele and then the CAD-FRED score. And again, this would be written as a tab delimited file. So there are 3 different scenarios depending on whether the ancestral allele and the CADFRED scores are annotated in the info column. So if you remember here, we have a checklist of information that needs to be specified to the program. So for example, you can have a VCF file that does not have the ancestral allele annotated in the info column, but it has everything else and it has the CADFRED score that is annotated in the info column. So what you would do is you would extract the fields here, and then you would use VEP to obtain the ancestral alleles for each SNP. So I'll go through what these files look like after we've gone through this table. So the second scenario is if we have all of the fields in the VCF file, but the CADFRED score is not annotated in the VCF file. So in that scenario, you would use VEP to annotate the CADFRED scores. And lastly, a third scenario when you have neither the ancestral allele nor the CADFRED scores annotated in the VCF file, which is what I came across. So in this scenario, you would use VEP to annotate both the ancestral allele and the CADFRED scores. So let me show you what these scenarios would look like. So if I go to test files, okay, let me just open a reader. Oh, it's here. So the first scenario is if the ancestral allele needs to be annotated. So you would have this file. This would be your main file. Let me just open that up. And then this would be your VEP file. Okay, so as you can see here, this is the main file and it has all of the fields that you need that is mentioned in the checklist. So we have your chromosome position ID, chromosome position, and in this case we don't have the ID field because in the VEP file we have the uploaded variation. Next is the reference allele, alternative allele, CADFRED scores, and all of the allele frequencies for each population. Now in your VEP file, you would have your ancestral alleles that are written in the third column over here. As you can see, this is the location. So the location would be the chromosome number and the position, and that would help align these 2 rows together. So do you see here? Here, and here. And then you have your uploaded variation, which can sometimes be your DB SNP reference number. So this is what the first scenario would look like. Let's look at the second scenario where you would need to annotate the CADFRED scores using VEP. So let me just close this. So again here you would have all of the fields that are needed in the checklist except for your CADFRED scores. And here you would have the CADFRED scores in the third column. Okay, so let's just close that. Let's go to the third scenario in which you would need VEP to annotate both the ancestral alleles and the CADFRED scores. So this is what the input files would look like. As you can see here, this file contains a chromosome number and position, reference allele, alternative allele, and all of the allele frequencies. But it does not have the ancestral allele and the CADFRED scores. So now I'm going to go over how you would run this on the command line interface and the graphical user interface. So I'm not going to go through all 3 scenarios. I'm just going to pick this scenario here. So we'll just copy this command here and go to the terminal. Let's just cd. Okay, and then we'll paste. Alright, so this is what is printed as it's running. And as you can see here, if we ls, these are all of the output files. Now, if we were running the graphical user interface, this is what we would do. So we have a few examples here. And this is the scenario in which you do not have the ancestral allele annotated in the VCF. So let's just move this so you can see. The input file would be the VCF _no_aa.txt. So that would be located in desktop FineMAV test files, and then this over here. Open. As you can see here, this is the input file. Next we have the prefix, so we're just going to use this and paste that there. Next is the reference genome, so that's HG19. Next we have the VEP file, so we're just going to browse, go desktop, FineMAV, and go to this vep_aa.txt and open that. And let's run this. Alright, program completed successfully. So this is what will be written as it's running the program. The next optional flag we're going to talk about is the penalty flag, and this part covers what the penalty parameter is and when do you use it. So the second optional flag that we're going to talk about is the penalty parameter. So I'm going to show you what that is. So it is this optional flag right here. So if you recall, FineMAV is calculated by multiplying three things: the derived allele frequency of the population, the CAD-FRED score, and the DAP. And the DAP stands for derived allele purity. Now, derived allele purity is a measurement that shows how spread out the derived alleles are between the populations. If the value is 1, which is the maximum, it means that the derived alleles are found in a single population. So if we go to the wiki, it says here that the derived allele purity, which is the DAP metric, relies on the penalty parameter, which penalizes allele sharing. So the authors of this paper, which you can see here, calibrated the penalty parameter by running simulations to find the penalty parameter that would maximize the difference between the differentiated positively selected variant and the less differentiated nearby neutral variants. So again, you can read more about it in their paper here. So for this program, we took their calibrated penalty parameters as the default values. As you can see here, the default values are available for number populations that range from 2 to 7. So if you're comparing between populations that fall outside of this range, or if you want to play around with the penalty parameter for your data set, you can use the penalty parameter option. So I'm going to show you what it looks like. Let's use this test file here, because this test file contains all of the information that is in the checklist. Okay, so if you were to do this on the command line, you would just copy this. We would paste that, and then we would just add -p, which is the penalty flag, and then enter in an integer or a float. So let's say we'll just put 5. And then enter. Okay, so this is what it looks like when it's done running. So if you ls, these are all the test files. So if we open up the log file, you can see here that the penalty parameter is 5. If you want to run this on the graphical user interface, you would do the same thing. So the input file here would be your VCF. The input file here would be your VCF_allfields.txt. Then let's call it test run. And then we'll make the penalty parameter 5. Okay, and as you can see here, it's generated all the output files. The last optional flag we're going to cover is the chunk size. So for this part, we're going to cover what the chunk size option is and when do you use it. Alright, so if we run the help manual, you can see in the optional flags down below, it says -- chunk size, or you can also use -c and then specify an integer. So the chunk size option is used if you're running a large dataset and want to save RAM. It will run the dataset in chunks, and each chunk would read a specific number of lines. In this case, the default is 200,000. So we have a test file so we can demonstrate how this works. So the test file, I've put it here, and it's called the chunk_test_file.txt. So we're going to open that up and see what that looks like. Here it is, and this is what the file looks like. So as you can see, it has all of the fields that are needed for the program to run. If you look back at the checklist in the wiki page, it says that it requires a chromosome number, ID, reference allele, alternative allele, ancestor allele, cat-FRET, and allele frequency for each population. And that's exactly what this file has. And the allele frequencies are given for POP1, POP2, and POP3. Now, this file is different from the test files that you see in the repository. This one has 200 snips. So if we scroll all the way to the bottom, you can see over here that it says 200. All right, so let's just close this. All right, so in order to see how it works, we can go back to the folder, and we'll just run this first so we can see the flags that are needed. So right now we need to specify the input file, which is the chunk_test_file.txt. Here, the next part would be the prefix. So that would be -x, and let's call it test run. And lastly, we have the reference genome. That would be HG19. So for chunk size, as we mentioned, you can just put -c and then specify an integer. So you would have to estimate the appropriate size for your chunks, and this would depend on how much RAM you have available. So if it has 200 lines, 200 snips, then we could have 20 lines per chunk. So I'm just going to put 20 and see what happens. Okay, so as you can see here, this is the output that it's given, and it tells you what stage it's on when it's running. So as you can see here, because we've split them into 20 lines per chunk, so that would mean that there are 10 chunks. And as it's being printed out, it will tell you what chunk it's on. So it says 1 of 10, 2 of 10, 3 of 10, and so on. And it does this until it reaches the bottom 10 of 10, and then it will output the file. So let's ls to see that. So here we have the log file, and then this would be the file that contains the intermediate calculations and the FineMAV scores. And these three over here are the bigwig files, and that is how you use the chunk size option. Okay, so now we're going to try running the chunk_test_file.txt file in the GUI. So over here I have the GUI version, so I'm just going to launch that. Okay, so over here we have the input file, so I've put that here, the chunk_test_file.txt file. And so that's what you can see here. The prefix is going to be test run. The reference genome will be HG19. And then over here under the optional flags, I can specify the chunk size. So in this case, we can make that 20, and then we'll see what happens. All right, so as you can see, a program's completed successfully. And as you can see here, again, it would tell you the number of snips, and then it would break it down per chunk, and it will tell you what chunk it's on as it's running, and it goes all the way until chunk 10 of 10, and then it'll be done. So that is how I use the GUI version. Thank you for watching this video. If you have any questions, please feel free to comment down below, and check out some of the other videos that are in these cards right here.